So this video will provide you your notes for the first section of your cell cycle mitosis set of notes in your packet. What we're looking at when we're talking about the cell cycle is not, and a lot of students make a mistake here, it's not mitosis only, but it's all of the life stages within a cell. And so what we're talking about are really divided into two major stages for a cell here. So the first of those stages is what we call interphase. This is the time between divisions during which the normal processes are occurring. I like to call this the in-between time because it's the time in between divisions, and you can match up the I in between with the I in interphase. So the interphase is the time in between divisions. First phase of the cell cycle. You learned in your POGIL, interphase is really G1, S, and G2 all together make up interphase. The second then phase of the cell cycle is what's called mitosis. In your POGIL, they call this the M phase. And this is the actual process of dividing the cell to form the offspring or the daughter cells from division. Just background information here so that we understand the process. And so really the most important thing to keep track of during the cell cycle is the DNA. The organelles are going to be divided equally between the new cells. It's the DNA that we really want to keep track of. And so just some review here. DNA, remember, is the molecule that stores your genetic information. You call this your genes. In eukaryotic cells, DNA is found in the nucleus of the cell. And it's shaped, as you know, like a twisted ladder. And so there's two strands. And across those strands are the bases that make up the rungs of the ladder. And the whole molecule is made up of just 4 separate pieces, what we call nitrogenous bases. These are the monomer of the DNA polymer. And so those bases are adenine, thymine, guanine, and cytosine. And we abbreviate those A, T, C, and G. Adenine, thymine, guanine, and cytosine. And so where those are is those are making up the actual pieces of the ladder. So you stick together a bunch of A's, T's, C's, and G's, and you can make a whole molecule of DNA. And so this is just looking at the ladder again, the DNA molecule, and what's called the base pairing rules, the base pairing rules of DNA. And that's that adenine always pairs with thymine, A always pairs to T, and that cytosine always pairs with guanine. So C always pairs with G. And so if you look on the ladder, you find, like right here, here's an A paired with a T, and over here, here's a C paired with a G. Okay. Now, what is an individual nucleotide? Okay, or the monomer. The nucleotide is one of the letters, so it's G. Okay. It's the sugar, and it's the phosphate group that make up the backbone. And so what I circled here, that's one nucleotide or one individual subunit of DNA. So what has to happen during cell division is that the cell needs to make copies of its DNA to pass on to the new cells, the cells that it's going to be making. And so DNA it's pretty unique in the way that it's copied, which I find to be really sort of interesting here. And so what DNA does is it needs to make a copy of itself. And so we start off with 2 strands, and I'm going to kind of follow with colors here. So we start off with, let's say, a red strand and a blue strand. Okay. Now red and blue in the beginning made one molecule of DNA. Okay. But what happens is during replication is we split the red and blue and we make a new half. So we'll call them the green halves to make complete molecules. And so basically we split the ladder in the middle and copy each side separately. And the DNA actually serves as the directions. So the blue strand are the directions to put together the new green side. And the same on the other side. The red strand are the directions to put together the new green side on this other side. And so you're turning one molecule into 2 by basically just splitting it in half and sticking in the bases that would pair. So if you have an A on one side, you're going to have a T on the other side. If you have a G on one side, you're always going to have a C. So we're going to move on from talking about replication to talking about some different structures within DNA, ways that DNA are kept in the cell. And so during interphase, the time in between divisions, you'd find your DNA in the form of chromatin. And so that's a DNA that's not wound tightly. It's not wound tightly. And basically, the purpose of chromatin is to provide the information or the directions to direct cell activities. And so it's loosely found inside the cell, kind of like a big plate full of spaghetti. The other form that we're going to talk about in mitosis are the chromosomes. And so these are going to be found during the M phase or during division. And so what happens is the DNA is really hard to sort of distribute during division. And so your cell is going to compact it and make it more easily transported between the cells. And so what your cell does is it coils that DNA into compact structures, and what it uses is a molecule called a histone, which is kind of like a globular protein. So if the blue is the histones and the red is the DNA, what happens is your cell takes your DNA and coils it around the histones, right, to make the strands shorter and more compact so that they can be easily distributed between different cells. And so this is just a picture showing the two different forms. So up here we have chromatin, and notice it's very sort of all over the place. It's not organized. It's all jumbled up, and it's loose. It's close to, single-stranded. Over here we have chromosomes, okay? And so what's happened is those individual strands have been taken and looped back on themselves, over and over and over and over again until they get very tight and compact and much easier to organize. And so kind of showing how that might be happening with the histones by looping it over and over and over again in order to make this X-shaped structure. Now, chromatin is found during interphase. Chromosomes are found during the M phases of the cell cycle, and only during the M phases. The DNA cannot be read during this time, so it can't direct the cell's activities when it's coiled. So this is just another look, sort of going all the way from chromatin, showing the histones, and then coiling into the actual chromosome during division. So like I'm saying, so this is interphase over here. This would be mitosis over on this side. Just a nice diagram. And so what happens is during division, we've replicated the DNA. So remember, we split it in half and copied each side. And so what we have are those chromosomes are X-shaped, right? And what that actually is each side is an identical copy. So if this side is capital letter A, this side is also capital letter A. They're containing the same information. And so down each side, we have what we call sister chromatids. Okay? So they're the exact same DNA, just being held next to one another. And so eventually, when the cell divides, each new cell receives just one chromatid, which is the complete set of information. The chromatids are held together at the middle by a little piece of compressed DNA and protein that we call the centromere. So that's the piece at the middle of the chromosome. So the whole thing is chromosome. Each side is a chromatid, and the middle is a centromere. This next picture will show that even better. So remember, each side here is identical. So if gene A is found here, gene A is also found down there. And if gene C is found at the bottom, gene C is also found over here at the bottom. So each side here is a sister chromatid. The whole thing is a chromosome, okay? And this part in the middle is the centromere. And that's basically, it's important to know and be able to distinguish between those different parts. Adding a couple more just pieces of the cell cycle. We've got some proteins at work helping the cell to divide. And the first one up is the spindle fibers. So you're going to see some pictures of of the cell division, right? And spindle fibers are basically like ropes or chains made out of microtubules that basically push and pull the chromosomes around the cell. The other part are the centrioles, okay? Centrioles, in green down here, are the anchor points for the spindle fibers. And so they're where the spindle fibers are attached so that they can push and pull on the chromosome. Okay, remember, we're calling this whole thing the cell cycle. And so the cell cycle includes the phases interphase, mitosis, and cytokinesis. So I, M, and C. Interphase, mitosis, and cytokinesis. So I like to say in between, multiply, and then cinch, or pinch in the middle, okay, which is what's happening in cytokinesis. And in the next presentation, we're going to be looking at this in a lot more detail. So you have to ask yourself, why are cells dividing? Why would they bother to make new copies? And there's really four main things that a cell is going to accomplish by dividing. First of all, asexual reproduction. So think bacteria, think single-celled organisms, making new copies of themselves. For growth, so we've talked about, a baby grows into an adult not by getting larger cells, but by adding cells. For repair, so let's say you have damaged tissue, okay? You get a cut or something like that. In order to repair that, what you do is you replace the damaged cells. You don't fix the broken ones. They die off. You grow new cells in your body to take up the area that those old ones occupied, or to replace, okay? So think of like a crab that loses a claw, okay, or starfish that loses an arm. So they can replace the claw or the arm by growing a whole new appendage. Now, not all organisms can do this, but some can. So we'll be revisiting this diagram, but I just want to show you, we've got the cell cycle here, right? We're starting at interphase. We're traveling in this direction through the G1, S, G2, and M phases in order to make two new cells. And we're going to get a lot more detailed with this right in the next presentation.